Hello, today I will be teaching you guys about the process of mitosis. In order to begin talking about the cycles of mitosis, we need to understand what mitosis actually is. Let's start first with the cell cycle. The cell cycle is a process where DNA must be copied, it must be separated, and the cytoplasm must also separate. It consists of 4 phases. You can think of them as a clock, where each quarter represents one phase. The phases are G1, S, G2, and M, where mitosis occurs. Mitosis is a stage in the cell cycle where the nucleus divides and so does the cytoplasm to produce 2 new daughter cells. Mitosis consists of stages within it, and I will discuss these in detail. The first stage is interphase. You can clearly see that there is a presence of a nucleus here, and within that nucleus there is uncondensed chromatin. A good way to remember this stage is to think of the chromatin being a ball of yarn. It is all in the nucleus meshed together and not separated. Next we have prophase, where the chromosomes condense. Now you can visibly see the chromosomes and not just them meshed together. It's kind of like separating a ball of yarn into 2 separate strands. You can also see the microtubules creating a spindle apparatus because they are about to start the fishing stage, which I will discuss more soon. The reason this is a 2n equals 4 cell is because there are 2 different types of chromosomes in this diagram, blue and red, and there is 4 total. In prometaphase, we can now see that the chromosomes have replicated and condensed. In this step, the main action is the microtubules fishing for these chromosomes, and by that, I mean getting in contact with them to start preparing them for the metaphase step. It is important to note that the nuclear envelope starts to disintegrate here because in the next few cycles, there is an absence of a nucleus. In metaphase, the chromosomes line up in the middle of the cell because the microtubules hold them in place there. You can think of metaphase as train tracks because train tracks line the cars up in a straight line, and that is exactly what happens here. In anaphase, the sister chromatids separate into daughter chromosomes because the microtubules exert a force on them that separates them. These daughter chromosomes go to opposite poles. That's why we can think of anaphase as a magnet. Imagine the top pole being positively charged and the bottom pole being negatively charged. The daughter chromosomes that attract to the top will be negatively charged, and the daughter chromosomes that attract at the bottom will be positively charged because opposites attract. This is just a way to put it into perspective that the daughter chromosomes go to opposite poles. In telophase, the nuclear membrane reforms and the daughter chromosomes go into the nuclear envelope. You can think of this as the daughter chromosomes on each pole go into their own nuclear envelope, and then the microtubules exert a force to keep these nuclear envelopes separate from each other. That's why you can think of this step as a magnet, where the nuclear envelopes are attracted to each pole but repel each other. In cytokinesis, there are 2 stages, the cell division beginning and the cell division complete. Here you can see that the cell membrane has been pushed in by a force, and that force is an actin-myosin ring that causes it to be pushed in. To simplify this, you can think of a finger being the force that pushes in the cell membrane. Finally, once cytokinesis is complete, there is 2 new daughter cells, and that is because they have divided. You can think of this as a pair of scissors being the force that cuts it into 2 cells, but in reality it's the force of the actin-myosin ring that causes the plasma membrane to be pinched in and eventually divide.